All right, so today we're going to walk through utilizing the Koji browser. This is a genome browser where we have a lot of ties into Cannapedia. So much of the data that was in this bioarchive preprint we have over here is now up in viewable in Koji. So this consists of, well, the paper had 42 genomes. There's now 47 genomes that are up in Koji, and that number will keep climbing. But these are whole genome shotguns done with Illumina, all mapped to a PacBio generated pan trio reference. A pan genome is just multiple genomes that build a reference together, and we have a maternal reference, a paternal reference, and an offspring reference, all assembled with Pacific Biosciences here. This is a long read sequencer that generates reference grade genomes, and the N50s of these genomes are very contiguous. The most contiguous genomes that have been put public. So N50 is the average size of the contigs in the genome, and the average for these are over 1 million bases. They're usually around 3 million bases in length, and that number goes a little bit further north if you get rid of some of the small contigs. So anyway, the important thing to know about these genomes is they have the highest Busco score out there. Busco is a tool that's utilized to measure the completeness of genomes. Okay, so that can be seen right here. Here's the mother, father, and the offspring. This is the N50 number, the average size of the contig lengths. And then, again, this is including a lot of small contigs. A lot of people like to screen out the things that are under 50 kb, and these numbers climb a little bit, but we've left them in there for completeness. You can see the genome sizes vary. The paternal genomes are bigger because they have a Y chromosome. This one's a female. And the Busco scores is the percent completeness by looking for around 2,400 or so genes that should be in all plant genomes. And so this is approaching the highest we've seen. I don't think we've ever seen this number in assembly go higher than 97.8. That's on a reference that we're working on right now. That's a little bit different than these, but nevertheless, this is the set of references we're using to map all of the other 40 genomes against this. Okay. Now, in addition to mapping those 40 whole genome shotguns against this data set, we have done isoseq, which is single molecule sequencing on PacBio of the RNA transcripts across 5 tissues. This gives us an idea of where the genes are. You can see we've got about 27,000 maternal genes and 32,000 paternal genes. The exome size is anywhere between 40 to 50 megabases. The coding size is a little smaller than that. So the CDS size is different than the exome size because the exome can have the 5' and the 3' untranslated regions. All right, these are UTRs. Okay, so when you make an RNA molecule, you often include the promoter and the terminator in the RNA, which aren't necessarily read into proteins. They're just used to regulate it. That's how the exome includes the CDS. So the CDS is always a subset of the exome. And then if you look at how much genetic distance these things span across, since genes can have large introns, that means these are the gaps in between the expressed regions, or the exons. This really spreads across 120 to 160 megabases in the plant. So anyway, a lot of data in there to chew on. I want to share a couple other points of this data set to you. This here is a copy number map of all of the other genomes that we sequenced against the Jamaican Lion paternal reference. Okay, so this paternal reference has Y chromosomes in there, and you can see there's 0 coverage over any of those contigs in all of these lines. And it turns out those ones all happen to be the males. These ones we know are female, and these ones all happen to be the females, and these ones happen to be the males. Male, female, and you can see there's coverage of those regions in the males, and there isn't any in the females. So that's how we identify the Y chromosome. It's actually a little bit bigger than this. There's some portions of the Y chromosome that are known as the pseudo-autosomal region, which is still recombining with the Y, which means it still has some high Y and X. Those are the portions of Y and X are still recombining, which means it still has some homology to the X chromosome. And then down here, you can see the females have twice the amount of coverage on these contigs, and they have they're normally covered in the males. So this is the putative X chromosome material as well. So The X chromosome probably entails some of these other areas as well. It's usually around over 100 megabases, and when we add up everything that's going on the Y's the largest chromosome as well. So just a little bit larger than X. That copy number map just helps you to identify which ones are definitively male and female. This is a pedigree that was sequenced, okay? So we did PacBio sequencing on this one, and this one. Now, this one is a runt. It's not apparent at this stage in its life, but you'll see some pictures forward that it's actually a runt, and we chose that as the offspring to sequence because we thought we might learn something about yield in that genome, since it seems to lack it. And then all the other ones we put through Illumina sequencing and mapped it back to the maternal or paternal reference, which you're going to see all of these BAM files in this CoGe database. Couple different aspects that you're welcome to read in this paper. These are the single nucleotide polymorphisms and the insertions and deletions under 50 bases that are found across all of the 40 genomes. We've got the type 1s, the type 2s, and the type 3 plants up here. The Jamaican Lion family is in here, and you can see there's less variation amongst their own family. That's because we're mapping the family back to its parent, and of course there's less variance that way. But as you begin to map hemp samples and other type 1 samples against these genomes, you start to see much more diversion and diversity. So, okay, so you will see VCF files inside CoGe that I'm going to show you that describe some of this data. This is a coverage correlation analysis of the whole genome sequencing data. Now, this is just here. Note the scale here. This is very, very highly concordant coverage across the genomes, but there's a couple genomes in here that are some of the most diverged genomes we've ever seen. This is the 83 set. This is a unique set of plants, minibisones, and they may have some hops heritage to them as well. There's been some really interesting crossbreeding they've been trying to do with hops. But most of the sequence we're seeing is predominantly cannabis, but you can see is, of course, the most divergent cannabis sequence we have here. So here's a picture of ADE. You can see its leaves kind of look like basil, not serrated, and it's powdery mildew resistant. Its neighbors that are loaded with it never seem to transfer it over to here. We've also seen some of that in Jamaican Lion family. They are powdery mildew resistant. We probably have some pictures of that we can share as well. This is JL5, which turned into being a hyper-branched runt when it came out of the growth chamber. It was grown in the same conditions as all of its siblings, which are towering over it in this photo. So there's some interesting things that we learned about what might be driving the lack of growth in that plant that you can read about more in the paper. So what you're going to see are a variety of heat maps that look like this throughout the paper. And these are largely heat maps looking at copy number changes of DNA in the genome. What we noticed in cannabis is that it loves to insert and delete copies of genes. In fact, this is how it did a cannabinoid gene expansion in the plant, as it just kept making more copies of these things, and that's why there's upwards to 90 of these genes in the genome that have what looks like an FAD binding domain and a berberine bridge, which is two of the characteristics of cannabinoid synthesis. There's a PFAM domain that searches for these in the genome, and that number ranks up there around 90. Now, not all of those 90 are active. Some of them are broken or truncated or don't have the proper start and stop codons, and so we've then filtered those for a smaller set of genes that you'll see that we're going to touch on in some of these slides. But nevertheless, you can do really interesting studies when you have sequenced whole genome sequencing data with very few PCR cycles. That's an important aspect of doing the method prep here. But when you do that, you can then throw the data up against the Jamaican Lion reference and ask which parts of the genome are missing, which genes are missing, and which genes are amplified. That's what you're looking at in this heat map here, is that the genes that are in red have been amplified quite a bit, and the genes that are blue have been deleted. So you can see already just in these set of samples here that there is kind of a characteristic of certain TLPs being deleted and certain chitinases and MLO regions being amplified. And also some chitinases that have been perhaps deleted or amplified. So now what we've been doing is tracking powdery mildew resistance status across multiple cultivars to see if any of these patterns map with resistance status. And the reason we're looking at chitinases and thaumatin-like proteins and MLO is these are all the genes in hops that have been hunted down over the years to be involved in powdery mildew resistance. So we already have a few candidates in here, genes that we believe are involved in the powdery mildew resistance in Jamaican lion, and we've cloned those and expressed them and demonstrated they actually destroy fungi. Now, that being said, while we think we have found a reason for powdery mildew resistance in Jamaican lion, we believe there's going to be convergent evolution. When there's this many chitinases and this many TLPs, other plants are going to probably find a collection of these things to amplify that give them resistance as well, but may not show the same pattern that we see in the Jamaican lion genetics. Okay, so there's multiple ways to probably skin the resistance cat, and while we think we found one of them, it probably may not be 100% reflective of resistance in other lines. Okay, so here's a set that we pulled out that we believe are playing a role. So we've cloned a few of these. In fact, this TLP 2563 has in fact been cloned. You'll see that work in this paper, that when it is cloned, expressed in E. coli and purified out of E. coli and then placed onto films that are growing penicillium and fusarium, we get this type of inhibition and growth when these chitinases and TLPs are present. Here's a fusarium oxysporum, and it's got less growth over here where we've applied some of the compound or some of the enzyme than we'll put down here. And it tends to change the autofusarin expression in fusarium. Now, fusarium is known that when you give it mycosides, it stops making these pigments. It's a sign of it going into apoptosis and cell death. So there's more to read about this in this paper, but if you really want to dig through this data, you want to get familiar with Koji, which is the genome browser that allows you to surf all this stuff. Now, we left off the last tutorial talking about CBD and THC genetics. This here is a heat map looking at those three genes that we scan in Canopedia, the inactive CBD synthase, the active CBD synthase, and the active CBD synthase. And you can see this type of pattern here that is inversely correlated and predicts the typing of the plants. The type 1, 2, and 3 status is completely predictable by the presence and absence of these genes. You'll notice this region is almost always correlated, maybe not here, there's a little bit difference, but the deletion sometimes extends to be a little bit shorter or a little bit longer in this region of the genome, and we get a more or less a very similar prediction with the gene even right next door to THC synthase. Okay, now, as I mentioned in the last tutorial, there's many other ways to knock out THC synthase. We have seen some type 4 plants that have this gene in red, making you think that it's a type 1 plant, but it actually has some point mutations that knock out really key amino acids. And I want to show you how you can take your VCF file from StrainSeq, load it into Koji, and look for those things, because right now the tools we have in the reports aren't digging that deep yet. The data's there in your sequencing data, but our software tools haven't gotten smart enough to go in there and identify the variants and decide which one's really damaging and which one isn't, to give you like a 23andMe report on every single base. We're building that right now, but in the meantime, we want to enable you to be able to load this stuff into the CoGe Genome browser and surf it yourself. Okay, so just to give you a sense of the complexity that's behind this, these are a very large set of cannabinoid synthase genes, 39 of them, with 11 other genes that are next door neighbors to them that oftentimes disappear or appear with them. They're in linkage. Okay, so when these genes get deleted, they sometimes delete their neighbors too. And that's important to track, because if you breed for certain cannabinoid synthase genes to be present or absent in your plants, you really kind of keep track of the neighbors it's dragging down with it. Okay, so this is a case here where these sets of genes, this is a gene on contigs, it looks at contig 756 right there, C756. This is the cannabichromene cluster. It's labeled as inactive THC synthase because that's what NCBI first learned of this as. It was submitted as an inactive THC synthase, meaning it makes very little or no THC synthase, but it might make some. And that's something to keep your eye on. It's now been cloned, and it's known to be the cannabichromene synthase cluster. Okay, so you'll see that many of the lines have copies of this. Particularly, some of these ones are in fact hemp lines. So when you breed for hemp lines, you got to make sure that if you have a hemp line that still has 8 of these inactive THC synthase genes, they may cause a problem. They may leak on you and still be slightly active, right? So a lot of growers right now want to breed hemp lines that have this 2 megabase deletion. There is a common deletion in the population where this whole cassette is knocked out. It also happens to knock out these other genes on contig 756 down here. Now, our paper describes these. These other genes here are in fact pathogen resistance genes. Okay, they're aquaporin receptors, they are gibberellin receptors. There's, they're important features that go with them. So you want to be careful and look at your plant to know if you are breeding for one of these things. You might want to pick one that doesn't have as many of those pathogen resistant genes knocked out. Like this one here, Tibor, still has many of those pathogen resistance genes, but it doesn't have the inactive THC synthase, so the cannabichromines are gone. So this is a great line to perhaps try and breed into a type 3 compliant hemp line. It may not make a lot of resin, so it may need to be paired with something that does. All right. You can also see several type 1 plants often have the cannabichromine cluster, and you would really never know if they're making residual THC because you already have a THC synthase and they're making plenty. But you will see a lot of plants that don't have the THC synthase gene do have a CBD synthase gene, but also have cannabichromine. That may be a problem, as we mentioned in our last thing. So anyway, there's a lot of cannabichromine genes to, a lot of cannabinoid synthase genes to keep in mind as you're doing these things and breeding, because ultimately these are going to be the source of pain. Residual cannabinoid expression is not just related to the major genes that are producing these things. Many of these old genes might be producing other cannabinoids, rare ones, or small amounts of THC and CBD synthase, which could wreak havoc in a hemp compliance program. So, okay, so feel free to find this paper at bioRxiv, and let's go to CoGe. So if you Google CoGe, CoGe, comparative genomics, you'll end up at a site like this. All right. There is a tutorial here, but I'm going to try and do that for you. There's a lot of genomes in here. You see there's 51,000 genomes in this thing. There happens to be a lot of our cannabis genomes in here too. So if you search for cannabis genomes, you're going to get a lot of them. You want to click on the one in here that's certified. So right down here, there's a Jamaican Lion mother that's been certified, and there is a male that's been certified. The certification just means that the Koji team has combed over the annotations and in the genome structure, and they believe it meets a certain standard, and they certify it. So these are the best Busco score and N50 genomes out there. You will note we have not put them into chromosomes yet. While we are playing with that, we're cautious on calling the chromosomes because they're all very similar sizes. And as we just showed you, there are frequent 2 megabase deletions that may make you miscall chromosome 5 versus chromosome 6 because these things are really close in size. And when there's a 2 meg deletion there, you can quickly make chromosome 6 mistake it for chromosome 5. I'm not saying they're on 5 or 6. And just as an example, there are a lot of these chromosomes are almost identical in size, and so the deletions matter. Okay, you may see chromosomes from us a little bit later this year, but for the most part, that level of resolution is a little bit academic at the moment. Let's open up one of these browsers. Okay, so this we're now into this genome. It will show you there's a couple different annotations that we've thrown in here. It will also give you a handful of experiments that are public and available for you to surf. If you want to download this genome, right there's a download button. There is a GFF download in here as well. We have had some people report that downloading from GFS from Koji changes the format a little bit, so just be aware of that. And there are 2 different ways to download it. There's like through this tab and one other tab that when you click on the actual experiment itself, I think when you download that, you get the exact file we put in there. If you download off of this, there might be some modifications that Koji added. So 2 different ways to download those, just be aware of it. Okay, so let's go browse this genome. And this is going to bring up what's known as a genome browser. And what it's going to ask you for are, what coordinates do you want to go to? What part of the map are you looking for? Okay, so here we are. This is that's probably the last thing we looked at. Let's just go over to, let's go to THC synthase. That's what everyone loves to see. 741. I happen to know this is at 441700. Is it 10? I think it is. I don't know. Okay. Where are we? Oh, there we go. Okay, so this shows you the A, T, C's, and G's. If you're not familiar with this, this is an amino acid translation in 3 different frames in the top strand and 3 different frames in the bottom strand. You can read 3 different frames of DNA when you're trying to convert it into proteins, and these red marks are stop codons. So usually genes don't have those, and so one way to know what frame you're in is just look for the longest stretch that doesn't have any stop codons. And with THC synthase, it's pretty easy. You can see there's a methionine right there. If you go further back to the start of the gene, let's zoom out a little bit. Let's not go in baby steps anymore. There you go. So start of the gene, stop of the gene. If you click on this, you'll get a little messenger RNA with a note of what the database thinks this is. We've done some annotation on this and adjusted some of the start and stop codons just to make sure that it's in fact what is seen in the literature. You can download the sequence of it by hitting that FASTA button if you want. It'll do something like this. There's the sequence. In fact, this is what we used to clone some of those powdery mildew resistance genes. We just clicked on the gene, grabbed this from the start codon on, sent it off to GenScript to get it cloned and sequenced and expressed, and we had protein in a few weeks. Okay, so what are you looking at here? This is, as I said, the coding sequence, CDS. If you want to add more features in here, you can easily take that CDS and it'll add it down here. You can remove that track if you want. You can remove that track if you want. Put it back, it'll end up down here. And then you can also see that we have all of these other reads here. This here is female leaf RNA sequence. Okay, this is when we did iso-seq sequencing, all of these RNA molecules popped out from the leaves that were expressing THC synthase. That's to be expected. This is a type 2 plant. Below here is a whole genome shotgun of grape stomper, another genome that's been aligned to this, and you can kind of comb through this and look for variants if you want. Let's get that out of here because it takes up a lot of memory and work with these. Okay, so here's THC synthase. Now, other things you might want to look at. Well, if you had your own sample, you could load that into the genome browser, a VCF file off of Canopedia, and see where all the SNPs are in your genome. And we're going to do that in just a moment. Let me just show you some of the other functionality here. Okay, so these are all the public data possible. You can hit the structural variation file here. We'll show you where the very large scale insertions and deletions are in the genome, and I don't think you're going to find any in Jamaican Lion over THC synthase, but you may find some other hits over here. Here's a deletion. Shows you its size and some of its attributes. Now let's go into THC synthase and let's ask ourselves, well, what do we have in the 40 Genomes project in terms of a VCF file? VCF files are variant call formats, and they'll have some of the SNPs. Let me see if that peppers this with variants. I don't think it does. Let me see. There must be another one up here that does that. We have a 40 Genomes thing public. Oh, there's annotations in here. This may give you some of the gene calls. Okay, it's kind of redundant. Oh, there is some work from a group, I think 20 Labs, that did some mass spec profiling, and you can see how many of these genes have they actually detected a mass spec peptide for. And in fact, THC synthase pops out. Now, in our experience, 98.8% of what Ben Orsborn has found and has published, we find in the RNA set in Jamaican Lion. But we only found he found about 12,000 peptides, and you saw from our slides over there that we have like 27 to 32,000 genes, and so we're only about 40% of his proteins are, or 40% of the RNA that we have found is accounted for in the protein data. That's not unheard of. Some of the proteins may not be heavily expressed, and then the sensitivity of the machine may not pick them up otherwise. So, all right, so we have a variety of other experiments here you can throw on top of this. We have the RNA from the female flowers, from the leaves, from the roots. Of course, there's not a lot of THC synthase expressed in the roots, as you can see down here. Let me just get more room here. So nothing from the roots. The seeded female flowers. Oh, RNA went down a little bit there. Not be surprised once you start putting seeds out. You can see the expression goes down on THC synthase. Not surprising. And then there's male flowers too. And the male flowers aren't making a whole lot of THC synthase. All right, I think that's probably not surprising to anyone in the field. The other thing we have in here are BAM files. These are binary after mapping files, okay, of about 50 or so strain-seq samples that are public in Cannapedia. So let's take a look at Queen Jesus. Now, Queen Jesus actually is not too bad. That one's a type 3 plant. Xanthica's a type 4 plant, so those aren't going to have a lot of sequence over this. This is a THC synthase, so let's hit something that's a type 1 so you can see what the sequence looks like on a type 1. Maybe Serious Happiness will be a type 1. So you can see there's some reads that are covering this region, and if we zoom in on those reads, you can see there's sequence coverage. Now, there is a region of THC synthase that is a little bit tricky to cover because it has homology to a lot of other cannabinoid synthase genes, and so it's hard to place reads in some portions of it. And sometimes the coverage might just be a little light there. Let's see if we get another one that's got a little bit more. There you go. This region's all tiled, but there's sometimes some lack of reads there in the middle. Let's pull up, maybe Liberty Haze will have some depth there. Yeah, you can get a lot of these. Now, if you go to something like Carmagnolia, I'm going to bet you won't see any reads because this is a type 3 plant and it is deleted in that region. Okay, so this is the underlying data that's behind the plots in Cannapedia. Now, if we did something like put a whole genome shotgun across this, I think what you'll find, these are whole genome shotguns, right? Let's find a whole genome shotgun that's a type 1 and see what we see. Reads tiled all the way across the region. Why? Well, whole genome shotgun takes the whole genome and sequences it. So we don't have to try to fish THC synthase out of the genome to get coverage over it with a hybridization capture system. We just sequence everything. It's a lot more expensive, but it's a lot more thorough. And it will miss less SNPs as a result of this, particularly in genes that have a lot of homology. Okay, so you can load up all different types of samples here and look for variants. Now let's talk about looking at variants. I think if you want to look at variants, we probably want to look at a VCF file. And all right, so now that we're looking at your VCF file, I'm getting a little more serious here and putting on a professional look. But in all seriousness, now what we need to do in CoGe is to actually load up your VCF file. And you have to have a Cyverse account in order to load data into CoGe, but it's really simple. Fill out an email address, you'll get a CoGe account in a day, and then you can upload your own data to this browser and overlay it with all the data that we have public. So the way you do that is you go over here to Experiments, you click New Experiment, or Load EXP, and this should bring up a window that allows us to load up a file. So we're going to hit the upload button here. We're going to choose a file, and you want to pick your VCF file. There's one right there. Now these can be a little big sometimes, so let's just see how well it soaks this up. We're in that range right now, and once this realizes this is the VCF file, you hit Next, fill out some meta information. Now I already have a version of this running that we can skip to while that thing is thinking. This is what you'll ultimately receive when you're done loading it. After the file's been loaded up, you'll have all this information, and it will collect some of the metadata. So some of the metadata it asked for, you can see in the background here. It asked for a name. You could put a description. We didn't put much in because it's just a test. A version, the site that it was generated to, whether it's restricted. We left it as private right here, so you can load your data in and you can view it against all the data we have in our database privately, and you don't have to put your put any of your findings public in this regard. So that's what that looks like. You'll see here, this is an important step. I've bounced over to this step here to make sure that's a VCF file. There's a lot of different files you can upload, but you want to make sure you call it what this is. And when you go next, it'll ask, I haven't really used these features yet. This will send you an email when it's done, but it only takes a few minutes for a VCF file. That's more important for BAM files. It can take an hour. And there's notebook features in here that I've not yet explored. And then when you hit next here, it's going to start uploading. I'm just going to actually cancel out of this because we've already done that section over here, and then just hit OK. So now that we have that loaded up, what we can do is look for it in a lot of the data that's up here. In fact, I'm just going to search for it. I think it's there we go, right there. So that's the VCF file we loaded up. We're going to hit View Data. Oh, we can even make it public just like that, real easy. Hit Browse, and it will probably load up the genome with this VCF file layered against it. Now, it's a variant call format. That just means every line is a list of a coordinate in the genome with a particular base change and some quality information about that base change. For instance, how many reads are contributing to that base change. So when you're zoomed really far out like this, you start to get a density plot of where all the polymorphisms are. Now let's go again to our favorite gene, 4417, 000. Now, if you don't recall 4417, I have that burned in my brain, but you don't have to have it burned into your brain. You can just go up here and say, I like THC, tetrahydro, and search. Oh, I forgot to check all of these, and then hit OK, and it will come up with a list of all the different THC-like genes in the contig. And I'm going to bet that one's it, right? No, that's not it. That's it right there. That's the one that is the active THC synthase. Just kind of remember that by the coordinate. And let's see. Let's get some of this in play. We want to see what the genes are. Features, CDS, there we go, coding sequence. Squish that back, zoom this out, and what you're seeing here are different variants. C to T variant at this coordinate near THC synthase. You can see another one over here, another one over here. Right now, if you zoom out a lot in this, you'll see many more SNPs. And each one of those is a variant. All right, so that's how you load your data into CoGe. And there's ways to load FASTQs in here as well. So it's a very helpful tool. Allows you to search around the genome and look for particular key genes by looking through this feature function. There's also some other comparative genomic tools in here that people should know about. If you were to actually go back, there's actually a tutorial on how to use it, so I'm going to point you to the tutorial because that's probably a better way to do it. But there are other comparative genomic tools in here, given this is the Comparative Genomics Genome Browser. If you go up to, I think, tools up here, you'll see those, like Heatmaps, a very good tool to do genome-to-genome alignments. In fact, let's do one right now. You can take Heatmap and align. Now, it will only do alignments of genomes that are, we want to do Heatmap too, that have annotations on them, and there very few of them have annotations right now. Am I in Heatmap? Maybe it's Heatmap 3D. Let me see. Oh, that's crazy. I don't want to do a three-dimensional genome. Alrighty. Heatmap 2 we're on. Select organism. So if you just go here and go cannabis, it's going to bring you up a lot of different versions of cannabis. And let's take Jamaican Lion as organism 1, and then let's do the same over here. Cannabis. And let me see, no codings, let me see, they don't, this one, so CBDRX has annotations. Purple Kush does not have annotations yet, although they're in NCBI. This does not have annotations. Maybe that one does now. Okay. So there, let's put on CBDRX. Generate SID map. Now this is going to run a program in the background, and it will get back to you with the map. Now that probably doesn't look very good to anyone here, so there are some features in here that will make this compare them in a more meaningful way. If you want this at 24 and 4, and I think the most important feature, you know, yeah, tell the chromosomes are 10. That's a let's do some KNKS. That's always fun. Let's give that a shot. Now, if you've run an analysis on here, it will remember that and only run the piece of your analysis that's needed, like this DAG chainer right now. If it's been run before and you happen to compare the same genomes side by side, it'll just recall the old time that you ran that. It has a running memory of the analyses that you've run in the past. But if you change any one of these numbers, it's going to have to rerun it. That will take a little time. Close out of some of these now. And we may have to come back to that at a later time. But this usually gives you some dot plots comparing one genome to another. And you'll see some of these in our manuscript, actually, so we don't have to wait for this. But they're actually in one of our figures where you can see Jamaican Lion lined up to CBDRX. And it just gives you an understanding of which contigs in our genome are the corresponding contigs in their genome. And it actually gives you an Excel file that you can use to download if you ever want to do comparative genomics between the two different genomes. All right, so lots of good tools in there, and we'll leave it at that, and probably put in another tutorial on some other functionality on this as video 3. Thank you.